Hello there, how are you? Welcome to another biology class. Today we will talk about the genetic blueprint of organisms called DNA. Before we start, make sure you have subscribed to this channel and please be kind to like and share this video. Alright then, let us begin.
DNA, deoxyribonucleic acid, DNA is a polymer composed of two polynucleotide chains that coil around each other to form a double helix. The polymer carries genetic instructions for the development, functioning, growth and reproduction of all known organisms and many viruses. DNA and ribonucleic acid, RNA, are nucleic acids. Alongside proteins, lipids and complex carbohydrates, polysaccharides, nucleic acids are one of the four major types of macromolecules that are essential for all known forms of life. The two DNA strands are known as polynucleotides as they are composed of simpler monomeric units called nucleotides. Each nucleotide is composed of one of four nitrogen-containing nucleobases: cytosine, guanine, G, adenine or thymine, T, a sugar called deoxyribose, and a phosphate group.
The nucleotides are joined to one another in a chain by covalent bonds, known as the phosphoester linkage, between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone. The nitrogenous bases of the two separate polynucleotide strands are bound together, according to base pairing rules, with T and C with G, with hydrogen bonds to make double-stranded DNA. The complementary nitrogenous bases are divided into two groups, pyrimidines and purines. In DNA, the pyrimidines are thymine and cytosine. The purines are adenine and guanine. Both strands of double-stranded DNA store the same biological information. This information is replicated when the two strands separate. A large part of DNA, more than 98% for humans, is non-coding, meaning that these sections do not serve as patterns for protein sequences. The two strands of DNA run in opposite directions to each other and are thus antiparallel. Attached to each sugar is one of four types of nucleobases, or bases. It is the sequence of these four nucleobases along the backbone that encodes genetic information. RNA strands are created using DNA strands as a template in a process called transcription, where DNA bases are exchanged for their corresponding bases except in the case of thymine, T, for which RNA substitutes uracil, U.
Under the genetic code, these RNA strands specify the sequence of amino acids within proteins in a process called translation. Within eukaryotic cells, DNA is organized into long structures called chromosomes. Before typical cell division, these chromosomes are duplicated in the process of DNA replication, providing a complete set of chromosomes for each daughter cell. Eukaryotic organisms, animals, plants, fungi and protists store most of their DNA inside the cell nucleus as nuclear DNA, and some in the mitochondria as mitochondrial DNA or in chloroplasts as chloroplast DNA. In contrast, prokaryotes, bacteria and archaea, store their DNA only in the cytoplasm, in circular chromosomes. Within eukaryotic chromosomes, chromatin proteins, such as histones, compact and organize DNA. These compacting structures guide the interactions between DNA and other proteins, helping control which parts of the DNA are transcribed. Property as chemical structure of DNA. Hydrogen bonds shown as dotted lines. Each end of the double helix has an exposed 5-phosphate on one strand and an exposed 3' hydroxyl group O on the other. DNA is a long polymer made from repeating units called nucleotides. The structure of DNA is dynamic along its length, being capable of coiling into tight loops and other shapes.
In all species it is composed of two helical chains, bound to each other by hydrogen bonds. Both chains are coiled around the same axis, and have the same pitch of 34 angstroms, 3.4 nanometer. The pair of chains have a radius of 10 with circle 1.0 nanometer. According to another study, when measured in a different solution, the DNA chain measured 22 up to 26 angstrom, 2.2 up to 2.6 nanometer wide, and one nucleotide unit measured 3.3 angstrom, 0.33 nanometer long. DNA does not usually exist as a single strand, but instead as a pair of strands that are held tightly together. These two long strands coil around each other, in the shape of a double helix. The nucleotide contains both a segment of the backbone of the molecule, which holds the chain together, and a nucleobase, which interacts with the other DNA strand in the helix. A nuclear base linked to a sugar is called a nucleoside, and a base linked to a sugar and to one or more phosphate groups is called a nucleotide. A biopolymer comprising multiple linked nucleotides, as in DNA, is called a polynucleotide. The backbone of the DNA strand is made from alternating phosphate and sugar groups. The sugar in DNA is 2-deoxyribose, which is a pentose, 5-carbon sugar. The sugars are joined by phosphate groups that form phosphoester bonds between the 3rd and 5th carbon atoms of adjacent sugar rings. These are known as the 3' end and 5' end carbons, the prime symbol being used to distinguish these carbon atoms from those of the base to which the deoxyribose forms a glycosidic bond.
Therefore, any DNA strand normally has one end at which there is a phosphate group attached to the 5' carbon of a ribose, the 5' phosphoryl, and another end at which there is a free hydroxyl group attached to the 3' carbon of a ribose, the 3' hydroxyl. The orientation of the 3' and 5' carbons along the sugar phosphate backbone confers directionality, sometimes called polarity, to each DNA strand. In a nucleic acid double helix, the direction of the nucleotides in one strand is opposite to their direction in the other strand. The strands are antiparallel. The asymmetric ends of DNA strands are said to have a directionality of 5' end and 3' end, with the 5' end having a terminal phosphate group and the 3' end a terminal hydroxyl group. One major difference between DNA and RNA is the sugar, with the 2-deoxyribose in DNA being replaced by the related pentyl sugar ribose in RNA. A section of DNA. The bases lie horizontally between the two spiraling strands. The DNA double helix is stabilized primarily by two forces: hydrogen bonds between nucleotides and base stacking interactions among aromatic nucleobases. The 4 bases found in DNA are adenine A, cytosine C, guanine G, and thymine T. These 4 bases are attached to the sugar phosphate to form the complete nucleotide, as shown for adenosine monophosphate. Adenine pairs with thymine and guanine pairs with cytosine, forming a 2T and G to C base pairs.
That is all for today, folks. One last thing, before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Brooke Solomon, MSc in Genetics. We will see you in the next video. Goodbye!